The overall goal of this procedure is to reproducibly generate Mycobacterium tuberculosis-infected macrophage aggregate structures in 96-well plate format for drug susceptibility testing using a viability dye.
This method can improve the early drug development process for anti-tuberculosis compounds, which is hindered by the unproductive translation of candidate compounds into the clinical setting. The main advantage of this simple, inexpensive, BSL-2-compatible infection model is that it recapitulates physiologically relevant cellular penetration barriers reminiscent of tuberculosis granulomas. This produces drug susceptibility results more predictive of in vivo efficacy.
Begin this procedure by preparing green fluorescent protein expressing M. tuberculosis MT²6206, hereafter referred to as MTB-GFP, for infection. Keep in mind that this is an avirulent strain, so all work in the protocol described here can be performed in a Biosafety Level 2 facility. For each 96-well plate to be set up, centrifuge at 2.8 × 10⁸ MTB-GFP at 3,200 × G for 5 minutes in a swinging bucket centrifuge. After the spin, aspirate the supernatant and add 5 milliliters of rPMIC to wash the bacteria. Pipette up and down to resuspend the cell, then centrifuge again. After the second spin, pour off the supernatant and resuspend the bacterial cells in 7 milliliters of rPMIC, then vortex for 10 seconds. Next, for each 96-well plate to be set up, centrifuge 7 × 10⁶ THP1 human monocytic cells at 250 × G for 5 minutes. After the centrifugation, pour off the supernatant.